Hey sqaudians! Welcome again. Just in our previous videos, we answered some very interesting questions like: what is Major Histocompatibility Complex? What is its main function? What are its major types? And how it plays role in antigen presentation? In a very detailed video, under the title MHC and Antigen Presentation. Then we have another very interesting short scanimation that will answer your query that, what is Major Histocompatibility Complex, MHC, Class I? So, before watching this video, you can go to sqadia.com and watch those videos to clear your basic concepts. Here in this video, we are going to answer another important question, that is: what is Major Histocompatibility Complex, MHC, Class II? Well, just as we know, MHC is a region of genes or complex of genetic segments found on chromosome number 6, with more than 200 genetic loci. And these loci are classified into three main classes. That are MHC Class I, MHC Class II, and MHC Class III. So, this second yet most important division of MHC is going to be our topic of discussion today. The three main gene segments for MHC Class II are HLA-DR, HLA-DP, and HLA-DQ. And the protein products of these regions are called MHC Class II molecules. And these molecules are known for their role in antigen recognition, just like MHC Class I molecules. And once they recognize the antigen, they present it to respective subset of T cells. That's exactly similar to MHC Class I molecules. But here the difference is that these MHC Class II molecules present exogenous antigen, unlike endogenous antigens by Class I molecules. And this presentation is to the helper T cells or CD4+ T cells, but not cytotoxic T cells as by MHC I molecules. We will look at the detail of this role later in this video. But for now, let's look at the structural features of MHC Class II molecules. Just like MHC I molecules, MHC Class II molecules are also non-covalently associated transmembrane heterodimers. That means they are made of two different transmembrane polypeptide chains, that are alpha chain and beta chain. Both of these chains are encoded by genes within the MHC Class II locus at chromosome number 6. And the molecular weight of the alpha chain is approximately 33 kilodalton, and that of the beta chain is approximately 29 kilodalton. Here, each chain has two domains, an extracellular domain, also known as variable domain, which is responsible for peptide binding and presentation, and a cytoplasmic domain, also known as constant domain, which is involved in intracellular signaling. The extracellular domains are named alpha 1 and beta 1, and the cytoplasmic domains are the and alpha 2 and beta 2. In MHC Class II molecules, the special site for antigen binding, that is binding groove, is created by alpha 1 and beta 1 domain for the peptides binding. And in this binding groove, there are variable amino acid sequences responsible for polymorphic nature of MHC, and thus its binding with large number of antigens. But in comparison to MHC I molecules, the Class II binding site or groove for peptides is more open. so that longer peptides that are 30 amino acid residues or longer can fit into the site and overlap at either end. On the other side, there is the base that enables them to anchor over the surface of cells and is made by the alpha 2 and beta 2 or cytoplasmic domains of MHC2 molecules. But here, unlike MHC Class I molecules, MHC2 molecules are not found on all cells, but are found on specific cells, mainly APCs, or antigen-presenting cells. These APCs usually include dendritic cells, macrophages, or B cells. So once these MHC Class II molecules get attached at the surface of these APCs, they bind with the foreign antigen at the binding groove, depending on the compatibility between the amino acid residues at the binding groove and the peptide antigen. Then these MHC2 molecules at the surface of antigen-presenting cells present antigen to the T cells. The T cells involved here are the helper T lymphocytes or CD4 plus T cells. And thus these cells generate action against the captured peptide antigen. So till now we have learned what is MHC Class II genes and MHC Class II molecules, and what is the structural features of MHC Class II molecules. Also, we got brief idea about the primary function of MHC2 molecules. But now, let's have a closer look at this role of MHC Class II molecules. That is antigen presentation to helper T cells. Well, we know that MHC Class I molecules present endogenous or intracellular antigens to the cytotoxic T cells via Class I or endogenous pathway. We have gone through the details of this pathway in another video. But MHC Class II molecules present antigen to helper T cell via Class II pathway, which is also known as exogenous pathway. This pathway is named as Class II pathway because it is followed by MHC Class II molecules, and is also known as exogenous pathway, because in this pathway, the antigen that is presented by the MHC molecules is the exogenous one. Now what does it mean? Well, exogenous antigens are the foreign antigens that are derived from the source external to the host organism, or they are typically derived from extracellular pathogens such as bacteria, viruses, fungi, or parasites. So, now let's see how these exogenous antigens are processed and then are presented by MHC Class II molecules to the respective T lymphocytes via Class II pathway. This pathway allows specialized immune cells, such as macrophages, dendritic cells, and B cells, to capture, process, and present antigens derived from extracellular sources to helper T cells. Here we are going to learn Class II pathway in a stepwise manner. So, in this pathway, the first step is antigen capturing. Antigen-presenting cells, APCs, such as macrophages and dendritic cells, actively engulf and internalize extracellular pathogens, such as bacteria or parasites, through a process called phagocytosis. Now here, if you are interested to know the details of this process, then we have another amazing video for you, under the title Phagocytosis. But for brief revision, phagocytosis is mainly engulfment of the antigen within the cell. So, first the antigen, under the influence of chemical signal, comes near phagocytic cells and gets detected by them. And then the internalization process activates. This process is named endocytosis. where antigen gets internalized within a vesicle named endosome. Once internalized, this endosome gets fused with the lysosome, which is a membrane-bound organelle that contains many digestive enzymes. Upon their fusion, another vacuole is formed that is now called as phagosome. Now in this phagosome, there is the captured antigen plus the digestive enzymes. So, once this phagosome matures, it ultimately causes the killing of captured pathogen or presented over its surface for recognition by other immune cells. And thus the antigen is thrown out of the cell, and this process is called exocytosis. So, either by phagocytosis or direct receptor-mediated endocytosis, the antigens are internalized in the APCs. Then the next step is antigen processing. Once internalized, the pathogens or antigens reside within specialized intracellular compartments known as endosome. This endosome then fuses with the lysosome to give rise to another type of vesicle, called phagosome. We know that lysosomes contain a large number of degradative enzymes. So now, within these newly formed phagosomes, the antigens are subjected to degradation by enzymes, such as proteases, into smaller peptide fragments. These peptides are then further degraded to 9 to 30 amino acid residues. Now this is the size required for their attachment over the binding groove of MHC, so now we can say the antigen has been processed. So here the next step is MHC Class II biosynthesis and trafficking. This step continues side by side with antigen processing, where MHC Class II molecules, consisting of alpha and beta chains, are synthesized in the endoplasmic reticulum (ER). During this biosynthesis, a specialized chaperone protein is co-synthesized with MHC Class II molecules. This protein is called the invariant chain and it associates with their peptide binding groove. And then this MHC invariant chain complex is then transported from the endoplasmic reticulum to the Golgi apparatus for further maturation, and then ultimately fuses with the endosomal compartment that was already capturing the processed antigen, or amino acid residues of the antigen. Then in the next step, there occurs the invariant chain processing. After fusion with phagosomes, the invariant chain on the binding groove of MHC molecules is proteolytically degraded, leaving behind a small fragment known as CLIP, Class II associated invariant chain peptide. This CLIP fragment occupies the peptide binding groove of MHC Class II molecules, preventing unwanted peptide binding. Then another specialized molecule, called the HLA-DM, acts as a peptide exchange catalyst, facilitating the removal of CLIP and the loading of antigen-derived peptides onto MHC Class II molecules. Now the next step is peptide MHC complex formation. So, once CLIP is removed, the binding groove is free. Then, the antigen-derived peptides generated within the endosomal compartments bind to the peptide binding groove of MHC Class II molecules. Now, complex is formed and is need to be presented over cell surface. So, we name this step as surface expression and presentation. So now, the peptide-loaded MHC Class II molecules are transported from the endosomal compartments to the cell surface by fusion of the vesicle with the cell surface membrane. Then on the cell surface, the peptide MHC Class II complexes are exposed, allowing interaction with helper T cells. Then comes the final step of this pathway, that is the helper T cell activation. We know that MHC molecules present antigen to helper T cells. So, like cytotoxic T cells, helper T cells, or CD4 plus T cells, also possess T cell receptors, TCRs, on their surface that can recognize and bind to specific peptide MHC Class II complexes. So, when a helper T cell encounters an APC, displaying a peptide on MHC Class II molecule at its surface, then the TCR of helper T cells bind to the peptide MHC complex. And this interaction triggers signaling events within the helper T cell, leading to its activation and the initiation of immune responses. So, all in all, by presenting antigens derived from extracellular sources, the Class II pathway enables helper T cells to recognize and respond to a wide range of pathogens. This interaction then plays a crucial role in regulating immune responses against foreign pathogens. So, I am very confident that now you can answer it very well. That what is Major Histocompatibility Complex, MHC, Class II? And we are here to answer more such interesting questions, along with astonishing visuals to clear your immunology concepts. So keep watching this immunology series, and keep visiting sqadia.com.
From medical students to seasoned professionals, sqadia.com offers engaging and dynamic content for your educational journey.